So in this topic, we're going to be going over how cells divide, or cell division. And in our first section, we're going to describe the process of binary fission, which occurs in prokaryotes. So here, bacteria are going to divide by a process called binary fission, and the process is modeled here in figure 10.1. It's not a sexual life cycle in that you don't have two parents combining genetic information. And so when the cells do divide, you're going to get clones. So this is, they're going to be, the two new cells or daughter cells are going to be genetically identical. Bacteria is going to have a circular chromosome instead of linear chromosomes like we see in humans and other eukaryotes. But it has to be, before any cell divides, you have to replicate the chromosome. The chromosome has the genetic instructions, has your DNA in there. So replication here is going to begin at a structure called the origin of replication, and in the model over here, that's going to be in green. So that's right up there. So there's your origin of replication. And here you're going to have the enzymes that replicate or make copies of your circular DNA. They're going to start at the origin, and they're going to take off in both directions. So as the enzymes are making copies of the two strands, they're going to go and eventually meet on either side here at the site of termination, which is in red. So that's another structural term to know. And then once the chromosomes have been copied, they're going to be partitioned or separated into two new cells, and then a septum is going to form in the middle. So we'll see this process here in this diagram. These diagrams are nice because they're going to have these boxes that describe the process. So I'm just going to go ahead and walk you through it here with the images here. Here you can see the blue part here, or I mean the green part is the origin, and you have your two new copies being made, and the enzymes that are doing the replication are moving in opposite directions there. Eventually the enzymes meet each other here at the termination site, and they're going to be facing towards the inside of the cell here in the middle, and then your origins are going to be positioned at about this location here and here, which is about 1/4 of the way, and over here about 3/4 of the way. Which, if we were to separate the cell in half right now, that would be halfway in the middle of this new cell and this one here. So as the two have the two copies of DNA have been separated, the cell is going to elongate, and then a septum is going to form in a process called septation. And at this point here, you're going to have some proteins assemble. These proteins go by the name of FTSZ that are going to help organize this septum here. And then eventually the two cells divide in half, and this entire process is called binary fission. So if you have seen cell division before in a biology class, you probably remember the word mitosis. Mitosis does not happen in bacteria. Mitosis we're going to cover in the sections coming up. Mitosis is a division that occurs when you have a nucleus. Since we don't have a nucleus here, they don't go through mitosis. So looking at the septation process, the process in which divides the bacteria cell, you can see that they've tagged the proteins involved in septation, and they're glowing in this image here. So these are those FTSZ proteins organizing that septation to occur here. So structurally this is going to help pinch the cells in the middle, and this particular type of protein assembly, the FTSZ protein, is found in most karyotes, including the archaea, which are those, there's two groups of prokaryotes, the domain archaea and the bacteria. So that FTSZ protein itself, some studies have recognized that the structure is similar to proteins called tubulin. You might recall tubulin is a protein that we saw as part of the cytoskeleton, the tubulin subunits that make up the microtubules. So it's structurally similar. However, the role of the FTSZ protein is different than we see the tubulin in eukaryotes. Here, the FTSZ protein is involved in septation, whereas the tubulin plays a role in dividing the chromosomes themselves during mitosis. So the roles during cell division are different. And then remember that tubulin forms part of the cytoskeleton as well. That's that FTSZ protein. Now, when it comes to cell division, we just finished covering how it occurs in prokaryotes, the binary fission process involving that FTSZ protein. Now we have the different eukaryotes here in the rest of this image, and you can see there are some differences here. Instead of the FTSZ protein, we have tubulin, and in some protists, that tubulin is going to organize into these microtubules that are going to form this axis of cell division, and then we divide our copies of our chromosomes. So you can see the chromosomes being divided here to opposite sides, and then eventually the cell can divide. Notice that the nucleus, or the nuclear envelope, which you don't have in bacteria here, but the nuclear envelope stays intact during this time. So the chromosomes, they get copied. As we have to, we copy our chromosomes, and we've got to separate them. But all of this happens within the nucleus. So eventually the nucleus has to divide, and then the entire cell divides. In other protists, we're going to see a similar process. We have structures that help assemble the microtubules in some species called centrioles, which they're made of microtubules as well. You see the microtubules form here. They go through the nucleus remains intact, and then you're going to have some microtubules that actually attach to the chromosome. So that's a difference here between some protists and other protists. Here, the microtubules actually attach to a structure called the kinetochore, and then helps to pull or separate the chromosomes into the two new cells. And notice that the microtubules are assembled from outside of the nucleus. In yeasts, which are eukaryotes, they're simple single-celled fungi, the yeasts are. Here, we're going to have kinetochore microtubules that attach to that, but the assembly happens within an intact nucleus. So that's the difference here, is the assembly is starting inside the nucleus. Everything is happening in the nucleus. If we compare that with the prior one, with these protists over here, the assembly begins from outside and then moves in, the microtubule assembly. You're also going to have polar microtubules, which are going to help in separating the two poles that we see here. And then in animals, there's a big difference here, is that the microtubules are going to assemble, and they are going to attach to the chromosomes, but a big difference here is that the nuclear envelope disintegrates away. So that's a significant difference here. Now, the type of cell division we're going to be describing here is going to follow the animal cell model, which, by the way, plants also, the nuclear envelope disintegrates as well. We're going to be looking and focusing on this one here, but be aware that there is variation in how this process occurs. So we're now going to look at the structure of the eukaryotic chromosome here, and your learning outcomes are going to be to describe the structure of the chromosome, and that includes looking at some special proteins called cohesin, and then condensins, a group of proteins that help organize your DNA. You also are going to distinguish between homologs and sister chromatids, and your third objective is to contrast replicated and non-replicated chromosomes. Okay, so we're going to open up by looking at this color-enhanced electron micrograph of some chromosomes having that typical X-type shape. So we're going to understand the structure of what we're seeing here. Every species can have a different number of chromosomes. Humans, for example, have 46 chromosomes, and they are actually 23 identical pairs. You get 2 full sets of 23 from each parent, so one set of 23 comes from paternal and the other one from maternal. And If we look at some other species, species will have typical types of chromosomes. Yeasts, for example, are going to have 16 chromosomes. Drosophila, which is the genus for a fruit fly, has 8 chromosomes. Green peas or garden peas are going to have 14. Horsetails, which we studied when we were looking at plants, they're allies of ferns. Horsetails have 216 chromosomes. Frogs have 26. Humans, which we just mentioned here, 46 in 23 pairs. And then dogs have 78 chromosomes. So 78 would be typical for a dog, and you could have other species that have 78. So it's typical for a species, but not exclusive to that species. So other species other than yeast may have 16 chromosomes, but it is typical. Now, for any one of these species, if you have any extra chromosomes, which can happen, or missing chromosome, that's going to be disaster, usually fatal for the cell. Here is a picture of an arrangement of human chromosomes. We can see that there's 23 pairs here. This specific arrangement is called a karyotype, and karyotyping is useful in looking at seeing if you have a male or a female, if early in fetal development, if there are any problems with number of chromosomes, for example. What happens here is they take a picture of the chromosomes during a part of cell division when the chromosomes appear. We're going to go over how the chromosomes appear. And so they take a picture of them, and then they can arrange them in pairs based on their banding patterns and a location of a constricted area called a centromere. So you can see a centromere on pair number 8 right there. Both of them have that same centromere. You can see they have similar banding patterns along there. So you can pair them up based on their size and location. Number 1 would be the biggest pair, and 22 would be the smallest pair. And then you have a final pair called the sex chromosomes because they're associated with determining sex in humans. Here we can see we have a male here because that final pair, when you have a smaller chromosome called the Y and the X, you get it's going to be male. But if you have 2 X's, that would be female. Doing this arrangement is a karyotype. So this would be a typical karyotype for a male human. Here, and showing that when you look at the total number of chromosomes here, you have 2 sets. That's called a diploid number. So the diploid number for humans would be 46. The haploid number would be half of that number would be 1 full set of chromosomes, and that would be 23 in humans. And we're going to represent haploid with an N and diploid with 2N. So 2N for humans would be 46. 2N for dogs would be 78. Okay? N for humans would be 46. And N for dogs would be half of 78. So let's do the math there. That would be 39. All right, it's 18 now. And so when you have them in pairs, they're identical in the sense that each chromosome is going to have specific genes on there. There's thousands of genes on a specific chromosome, especially the large ones. You have thousands of genes on there, and the same genes for specific instructions are going to be on those chromosomes, right? So if you have a gene for, say, eye color on one of the chromosomes, the other chromosome in the pair is going to have that same gene on there, right? So those pairs are called homologous pairs. It's a vocabulary word. So they're homologous pairs. And each individual of the pair is called a homolog, right? So for, let's say, pair number 19 down here, this is your homologous pairs, and each one will be called a homolog of the other. So you take a homolog of number 19. So looking at the chromosome itself, the chromosome is where you find your DNA, but the chromosome is going to be complexed with proteins. So chromosome is not just DNA, it's going to be DNA with protein, especially proteins help arrange. Now, depending on the location of the DNA, you may see some complexes with RNA as well. because DNA is used to make RNA. So when DNA is actively making RNA, during the process we'll cover later, you're going to find RNA within that temporarily bound with the DNA in there. So that happens during RNA synthesis. So instead of having a circular DNA, eukaryotic cells have linear DNA. So the two ends are not attached into a circular pattern. So in humans, every one of the 46 chromosomes represents one long strand of DNA. And it's double-stranded DNA, just like we studied when we were looking at biomolecules back in an earlier chapter. And a typical human chromosome of the 46 is going to have 140 million nucleotides. So remember, each one of these is a nucleotide. Nucleotides make your nucleic acid. So imagine all along that strand, 140 million of those. So 140 million of these bases. Okay? And so in your nucleus of a cell, you're going to have it organized into something called chromatin. And so chromatin is an arrangement of both the DNA and the protein itself. And there are going to be two kinds of chromatin. There's going to be heterochromatin and then euchromatin. Okay, so the euchromatin is going to be the one that's actually going to be actively expressed to make proteins, and the heterochromatin is going to be the parts of the DNA that are not expressed. Not all genes are going to be expressed or be translated into proteins in a cell at a given time. So looking more closely at the structure, you can see the different levels from just your DNA here and then on to your highly condensed chromosome here. In fact, the matter is DNA is way too thin to be seen. The diameter of a strand of DNA is too small. It's 2 nanometers in size. You can't see it with a light microscope. And even at 11 nanometers, you cannot see it. 30 nanometers, you cannot. But once you start getting around 300 nanometers to 700 nanometers, it's now big enough to be resolved by a light microscope. And then you'd be able to see it there. So the DNA itself is going to be complexed with proteins called histones. Okay, so this is a group of type of proteins called histones. They have a positive charge to them, and that's attracted to DNA, which carries a negative charge. So when the DNA combines with the histones, it's going to wrap around it about almost almost two full times, about 1.7 times or so around. There's going to form these structures called nucleosomes. And so the nucleosome is going to be the DNA wrapped around it. So you can see here in this this part here, the histones are here in the middle, and the DNA wraps around that, and that one structure is called a nucleosome. Now remember, soma means body, and nucleo means bodies found within there. And then you're going to have short segments in between the two, called linker DNA, links one nucleosome to another. So it overall looks like beads on a string over there. So these nucleosomes occur about every 200 nucleotides. So you're going to have your maybe 150 or so nucleotides, and then the remaining linker, and then another 150 in that wrapping around there. So that's the next level of organization where you have the nucleosomes. And so after that, you start to get other proteins that are going to help organize this further. These are you're going to start getting into proteins called not the histones, but other proteins called condensins. And those are going to help organize that DNA. Now, during your typical cell, at any given point in time, they're going to be, they're often thought to be, the DNA is often thought to exist at about this level right here, where it's about 30 nanometers. And we can refer to that as the chromatin. And so at that chromatin, you can't see with a light microscope. So when you're looking at cells under a microscope, you're not going to be able to see the chromosomes because they're in the form of chromatin most times. So I want you to keep this in mind because we have to oversimplify it. Other things when we're talking about modeling the behavior of these chromosomes. And sometimes we'll draw them as chromosomes, but really they're going to be in the form of chromatin, which we couldn't actually see with a light microscope. So we had already gotten to about this level here that's already been described here, the 30 nanometers of the nucleosomes. The nucleosomes and the beads are only 11 nanometers, but you start to get the DNA to become even more structured and slightly more condensed. And so you get them into these 30 nanometer structures, and at that point they're called solenoids. So here is the chromatin is the form of solenoid. And then during cell division, when we make copies of your DNA, the 2 copies of DNA are each going to be in the form of chromatin, but they're going to, because the DNA is really long, we're going to have to condense it even further, and that means organize it even further by coiling it up. And so this coil-like structures are going to be further arranged or scaffolded with these scaffolding proteins, and those proteins are called condensins. And they fit a general category of proteins that are also seen in prokaryotes that help organize the chromosome overall. And so eventually you get to this chromosome that we saw in the karyotype. So that karyotype, or the picture of the chromosome, was taken right when the cell is going to divide. If the cell is not actively dividing, the DNA is going to be more or less about this level here. Okay, so most of the time the DNA is at this level. Only once we've made copies of DNA can you actually see the chromosome. Keep in mind that word chromosome has the word soma in it again, which means body. In this case, this is a body that picks up a stain. So when they treat the cells with stains and they first observe these, they can see them. And these bodies were observed, these colored bodies. Chromosome is colored. So here, this is the way your book represents it. This one was from another source, that image that you see on this slide. But coming here, you can see your DNA here. You have your nucleosomes, and then you start having the solenoids formed at this point. And at this point, you're about 30 nanometers in diameter. And then you're going to have these scaffolding proteins, which are part of those condensins. They're going to create loops along the scaffolding proteins there. And then eventually you get your chromosome here, that familiar X-shaped chromosome, with a constricted area in the middle here called the centromere. And this is only going to be during mitosis or cell division, so this is your mitotic chromosome. For the most part, DNA is thought to be arranged or organized about this level here in the form of what we would refer to as chromatin. So I mentioned earlier that before cells divide, the DNA has to replicate. Now, remember that DNA is associated with these proteins. And the DNA, when it's going to replicate, is not condensed like a chromosome. Okay? So it's not going to be like this. The DNA is going to be more loosely arranged in the solenoid or maybe even lower level than that. And it has to replicate, just the way we saw the bacterial chromosome has to replicate. Every one of the 46 chromosomes need to go through replication. So every one of them is going to go through replication before that cell divides. And then each cell needs to get one full set of those 46 chromosomes that are going to go in there. So if you think about it, you have only one strand. And that one strand is going to replicate. And it's typically in this form here. Okay. So it goes through, opens up, replicates itself, and the 2 copies there are going to stay stuck together. Okay? And that's going to be a key in how we're going to get 2 full sets in each cell. So the 2 copies actually stay stuck together, and then before they divide, they go through this condensing, where they get coiled up and get very, very thick. And when we get to this level here, they stay stuck right there in the middle. And actually, these 2 arms of the chromosome are the 2 exact copies of DNA. They're going to be held together there. So there's special names for this. So they oversimplify here. I said sometimes you're going to oversimplify and just call them chromosomes. But really, we're not in the stage of a chromosome. Okay, so really, this blue, what they're calling a chromosome here, is really uncoiled, and it's in the form of a chromatin. And then it has to, the DNA has to replicate itself. So really, we can't see. It's not condensed like this. Okay? But remember, we were talking about having homologous chromosomes. So let's say this blue one is one of the 23 pairs. So one has to come from mom, one comes from dad, one paternal, one maternal. And there is 22 other pairs just like this. So each one of the pairs is going to have a homolog. So there's one homolog and the other homolog. Okay. Each one of those has to go through a replication. So they're going to replicate, and when we go through that replication, you're going to end up with two pieces of DNA that are still stuck together, and the reason they're replicating is because the cell is about to divide. Okay? So when the cell is about to divide, they replicate, and then when we're going to divide, the chromatin now, with the two pieces of DNA still associated with each other, are going to coil up and make this X-like looking structure here with these two arms of the chromosome, and each arm is an exact copy, and we're going to call those sister chromatids. So that's another vocabulary word. Okay, so while this is an entire chromosome, this is a sister chromatid. Okay? And then here's the other homolog. Okay, so you're going to have 22 other pairs in a human cell. And what's going to happen here is the two sister chromatids are going to be held together by, I talked about condensins to help coil up these chromosomes into this more structured, highly condensed DNA. There's going to be another group of proteins called cohesin proteins. And the cohesin proteins are going to be what help hold the two sister chromatids together. And they're going to be held together there until the two sister chromatids get pulled apart. So think about this here, that when the cell's about to divide, we normally have 46 chromosomes or 46 pieces of DNA. Now we still have 46 chromosomes because we're still going to call this a chromosome, but really it's two exact copies of the DNA. So really, right before we're going to divide, you do not have 46 chromosomes. You have 46 times 2. You have 92 strands of DNA highly condensed into sister chromatids, and then during division, each chromatid, which is an exact copy of its DNA, is going to get pulled apart into two new cells. So the 92 chromosomes get pulled apart, and each new cell is going to get exact copies of the cell. Exact copies of the 46 chromosomes, I should say. So now remember that these chromosomes are only going to be visible during cell division. So some key terms here that we should be familiar with here is you have DNA, and the DNA with the proteins that initially help organize it somewhat. So it's the DNA and the protein called the histones. Okay? And when they coil up just a little bit more, we're going to refer to that as chromatin. And then from the chromatin eventually coils up to form a chromosome, but you don't get a chromosome until you got your DNA replication, and they actually coil up to look like what we saw in the karyotype. So in reality, during the normal cell activities, if the cell's not going to divide and replicate its chromosomes, we don't actually see the DNA like this. It's uncoiled and it's in the form of chromatin. So if we were to look at the cell cycle of a eukaryotic cell, which is the goal here for your learning outcomes, is to describe the basic stages. And we divided it up into one full cycle and made a pie chart out of it. We can cut a wedge out here in the middle and see that most of the time the cell is going to spend in that larger section there, from there to there, and that process is going to be called an interphase. And that interphase can be divided into 3 parts, and the largest part might be better if I made this way here is going to be called the gap 1 phase. And this is when you start off with a new cell. The cell is just divided. You have 2 new cells, and each cell is there during the gap 1. And then during that gap 1 phase, this is going to be where those new cells are growing, and they're going to be going about their normal activities. And it is the longest phase there. And at some point, a cell may be triggered either because it's gotten too big or by some external signaling from other cells to prepare for cell replication, in other words, or cell division. And in order to do that, recall that we have to make copies of our DNA. So that is going to be called the S phase, and that stands for synthesis. And what's going to happen there is we're going to be replicating every one of the DNA. Sorry, and sometimes we'll say every one of the chromosomes is going to replicate. Well, your DNA is really what's replicating in there, right? Remember, chromosome has a very specific meaning. And you only see it right when the cell's going to divide. So at this point here, we're going to replicate, and you're in the form of chromatin. And then once you've done that, you've committed to dividing. So you're already going to go divide. So you then enter gap 2. Okay. And during that stage, some of your organelles are going to be replicating, and you're going to start preparing for building those microtubules. So you're going to start making lots of tubulin. Remember that we talked about that in an earlier section, that the microtubules are going to help separate the chromosomes. In this case, we now know what we're going to separate. We have a name for it from the prior section called the sister chromatids, which are the exact DNA replications or replicates that were made. And so once we get there, we're now going to enter what's called the M phase. The M phase is also called the mitotic phase. And we're going to have, I'm going to just draw it as a P for prophase, and then M for metaphase, and then we're going to have an A for telophase, an A for anaphase, and then a T for telophase. And and these are going to be the stages that describe what's going on with the chromosomes. Remember, we can see the chromosomes here. And then at some point, usually starting back in anaphase, we're going to have the entire cell divide. So we're going to call that little arrow in there, that's going to be called cytokinesis. Sometimes the books and the diagrams are going to say cytokinesis happens when we're done with telophase, but cytokinesis, which is really the division of the entire cell itself, the cell pinches off in the middle, that's called cytokinesis, or movement of the cell and the cytoplasm, separating the cytoplasm. But you first have to divide the nucleus. So that's the mitosis, and what you're dividing as the chromosomes, and then cytokinesis occurs. So all of that combined, both the cytokinesis and mitosis itself, is part of the M phase there. And so again, mitosis, we're going to divide that in 2 phases. Your book actually gives an extra phase besides just the P and the M. The P stands for prophase. And then there's metaphase for the M. And then I mentioned anaphase. And then you have telophase, or telophase. Your book adds, sometimes the books refer to it as late prophase, sometimes they refer to it as prometaphase, which means just before metaphase. But the way I've always remembered it is I had an old biology teacher in high school said, just remember IPMAT. And then you have to remember cytokinesis occurs. The I is for interphase. This is the inter means between, so this is the stage between the cell divisions that occur. And by the time you're done with cytokinesis, you're going to have 2 new cells, and each one of them goes into gap 1. And then we can track one of those cells. Okay, so this is the way your book draws it here. Here's your gap 1. Your synthesis is in red, and then gap 2. And then the purple is the mitosis, and then they have the blue for cytokinesis. But remember, cytokinesis will actually start back at anaphase. So how long does this whole cycle take? Well, the answer is it depends. If you're a very early embryo in early embryonic development, Sperm fertilizes the egg, cells going to be dividing rapidly. And so that can be as short as 8 minutes, like in fruit flies, right? Or every 20 minutes in other embryos. And for mature cells, a mammal cell may take 24 hours. They need time to grow, get through gap 1, and then if they're signaled to begin to divide and replicate that DNA. Some cells, like your liver cells, can take more than a year once they're signaled to start going through and commit to dividing. And during that entire year, most of the year, they're going to be in gap 1 during that whole time. So the longest stage of the cell cycle is going to be interphase, and in interphase, the longest stage is going to be gap 1 overall. Well, if a cell commits to doing this, it's going to take a little bit of time for them to synthesize their DNA and then do the final preparations for gap 1. The entire M phase, though, will only take about an hour. Okay, so it depends. Now, some cells are going to stay permanently in gap 1, and we're going to call that gap 0. Other cells will stay in gap 1, and we're still going to call it gap 0, like a liver cell, until they receive some cues signaling that says we need to divide, and then they'll get into and complete gap 1 and then start S. Some cells, like your nerve cells and your muscle cells, will stay permanently in gap in G0 and never go through division. So it depends on the cell type. So here we're going to focus specifically on interphase, which can be described as preparation for mitosis, but this is once you get into the S phase part of it. So your learning outcomes here are going to be to describe the events that take place during interphase and then illustrate the connection between the sister chromatids after S phase. Okay, so what is their relationship at that point? And so looking at interphase here, remember we have G1, S, and G2 phases. In the G1, this is where you have a lot of growth and the cell goes about its normal business of doing the functions of that cell. At some point, though, the cell moves from G1 and goes into S because of some either internal or external signaling that says we're going to divide. And so that gets into S, or synthesis, to replicate the DNA. And then G2, you're going to have some final preparations. We're going to have tubulin synthesis, centrioles, which were mentioned back in section 1, are going to replicate. The tubulin is going to be used to form microtubules, which you can see in the image here. There's these green strands here are your microtubules. And the chromosome during G2 is going to start to condense and coil up because it's in the form of chromatin up to this point. But once you're in G2, we're going to coil up, and once the chromosome has become highly condensed, we've now going to enter the prophase part of it. So during G2, we start that coiling up. Now, once the chromosome begins to coil up, each one of so remember that during S we had copies of DNA, and so we look at a human cell, you have 46 total chromosomes. So this is just one of them, one of a pair of homologs. And each one of these is a sister chromatid. Each one of those is going to have this constricted area here, which is called the centromere. So each one has a centromere, and the two centromeres are going to be held together in the middle by a cohesin protein. Okay, and at the centromere area, you're also going to have these plaque-like proteins called kinetochores. And kineto comes from the word kinesis, which means to move. We saw a whole study area study called kinesiology. There was kinematics. They took some basic physics class. That kinesis means to move, and it's that this kinetochore is going to be, each one has its own kinetochore, is going to connect to the microtubules, and you're going to call those kinetochore microtubules. That's the point where they attach there. So all of this is going to occur for each one of the chromosomes. They're in pairs, sister chromatids. They're held together by cohesin, and then the microtubules connect at the kinetochore, and they have a special name now. The protein that helps hold them together is kinesin, but in metazoans, which is a fancy word for animals, the cohesin may be replaced by the condensin-type proteins that help do the organizing of the chromosomes to begin with. So they are showing here a picture of a metaphase chromosome, which is the point at which the chromosomes are most tightly coiled. But the condensing and the formation of the chromosome itself, where it becomes very, very thickened and highly condensed, the DNA in the form of the chromosome, starts to begin in G2. So in G2 is coiling up, condensing, and then once you're able to see it, we call that the prophase. And so this is the first image of a series of diagrams that take us through the process here. And this would be an interphase during G2. You can see that the centrioles have replicated, so we have paracentrioles here and here. They're going to start to move to opposite sides of the cell. You're going to start to see microtubules begin to form and organize. In here, we're still in interphase. You cannot see any chromosomes. It just looks real grainy in there within the nucleus. We don't see chromosomes just yet. So we're still in interphase at this point, but we're heading towards prophase. So this is, we're looking here at interphase G2 when you start to see the centrioles separating to either sides of the pole. So here in this section, we're going to be looking at the mitotic phase. And it's going to be a phase where those chromosomes are going to segregate or separate, and we're also going to divide the cytoplasm. That process we talked about called cytokinesis. Your learning outcomes here are going to be, you should be able to give a basic description of mitosis, and that includes knowing the basic structures associated with mitosis, as well as what's going on, the changes that are occurring within those structures. So mitosis is going to be divided into 5 phases. In the earlier section, I mentioned just prophase, metaphase, anaphase, and telophase. And then there's prometaphase, which means before metaphase. Sometimes that's called late prophase. And so we'll start with prophase here. And prophase, recall that if this is in animal cells, plant cells don't, most plant cells are not going to have the centrioles that you see here, but they are located within an area called the centrosome. That centrosome and the centrioles are going to replicate, and these structures that began to separate and move to either ends of the poles there at the end of G2, they're going to continue that process, but microtubules are going to begin to start to assemble. So you can see these green structures in this image. They're colored green here for us to distinguish from others, but those are your microtubules that are beginning to organize. You're also going to have microtubules that are actually moving outward towards the plasma membrane. Those are going to be called asters, and they're going to help support these structures. As these structures move to either end of the pole, they're going to brace themselves against some of the cytoskeleton in there because the other microtubules that attach to the chromosomes are going to end up pulling apart the chromosomes. So the asters are thought to help brace or hold these polar structures on either side of the cell that's going to eventually divide. Another big thing that's going to happen here, you can kind of see that purple nuclear envelope is going to begin to break down or disintegrate. Okay, so, and this whole time, the chromosomes are continuing to condense tighter and tighter. They're going to be the most tight at when we get to metaphase. So that process of condensation is going to continue. Then we're at prometaphase, or sometimes called late prophase. So prometaphase here, this is going to be a key point here is that your microtubules, which are part of the spindle, the mitotic spindle, are going to connect to the kinetochore. So we're going to call those kinetochore microtubules. So there's your kinetochore on either side. So each one of the chromosomes now is going to be attached to both poles. Remember, this entire thing is called a chromosome, but each sister chromatid, one's going to be attached to one pole and the other chromatid, the part of the identical pairs, are going to be attached to opposite poles. Now this is oversimplified, so let's say that this chromosome and this one, those are the homologs, or the homologous pairs. Each one has a sister chromatid, a pair of sister chromatids. So the sister chromatid's identical copies are going to be pulled to either side here. In this simple model here, this one would have a diploid number of 4. So there's 4 chromosomes, and there's only 2 pairs of chromosomes, but the sister chromatids are replicates of each one of those homologs. So that's a key point you don't want to get confused about, is that each one of these chromosomes contains identical copies of that DNA. So what's going to happen here is, once the attachment occurs here, these microtubules are going to be assemble each other and disassemble. So they're going to break down, build back up, and that's going to cause a lengthening and shortening of each one of these kinetochore microtubules. And then there's also going to be motor proteins that actually can move along the microtubules. So the combination of the assembly and disassembly of the chromosomes, that's going to cause this sort of a tug of war on those chromosomes. They're going to move back and forth until eventually they get to the middle. When they get to the middle, we've gotten to metaphase. Okay, so that's the key thing here is remember, meta means middle, and here their chromosomes have lined up perfectly along, and it's really more of a two-dimensional plane. So if we were to just if you look at it from a certain angle, it's going to look like the chromosomes are all along a straight line, but they're not. If we just look a little off center here, you're going to see that the chromosomes are actually lined up around a two-dimensional plane here. And so we call that two-dimensional plane. It doesn't really exist, but we're going to call that plane the metaphase plate. It's kind of like the equator on the Earth. It's not really there in real life, but we'll draw it on a globe. And so this is going to be the point from which, once you get to that point, the next stage is where we pull apart the sister chromatids. And so the key here for metaphase is that the microtubules have been are all lined up now in the middle on the metaphase plate. I should mention here, there's going to be 2 kinds of microtubules in the spindle apparatus, or the mitotic spindle. There's going to be the kinetochore microtubules that connect to the kinetochores of the sister chromatids that you see here and here. There's going to be another set called polar microtubules, and they don't connect to the chromosomes. They simply reach out toward either pole, and there's actually one from each pole, and there's several of them all along here, and they're going to overlap with each other, and they're going to play a role in helping to stretch out the cell later. So there's 2 kinds of microtubules. There is the kinetochore, and then there is the polar microtubules. And then don't forget we have the aster out here, which is helping to brace. They're also made of microtubules. They're going to help brace these polar structures there to give support to so that the sister chromatids get pulled apart. And then we move on to anaphase. Anaphase here, the cohesin, that protein, or the condensins that were holding the sister chromatids together, is going to be broken down in every one of the chromosomes, and all simultaneously at the same time. So this is a mechanism of control there. When the chromosomes are held there in the middle at metaphase earlier, they're under some tension. They're already being pulled on. And at that point, when they line up in the middle, an enzyme is going to be activated. It's going to go and simultaneously dissolve the cohesins, or the condensins, that are holding the sister chromatids together before they get, so that once those proteins are broken down, the sister chromatids can begin to be pulled apart simultaneously. So every one of them begins to be pulled apart at the same time. There's going to be two types of anaphase. There's going to be the anaphase in which the sister chromatids are being pulled apart. So the kinetochores, there's a kinetochore in each sister chromatid. They're going to be moving, the kinetochores are going to be moving and taking the rest of the chromosome with it to either pole. They were sister chromatids, but now that they're separated, they are separate bodies. We can start referring to them as individual chromosomes again. The other kind of anaphase is going to take place is anaphase B. And here, this is where the polar microtubules, you can see they're overlapped right here. There's overlapping of these polar microtubules. There's going to be motor proteins that are along there, and those motor proteins are going to be sort of doing this movement that's going to cause these polar microtubules to push away from each other. And what that does is it's going to separate or stretch out the cell further, so that we can get those sets of chromosomes as far apart as possible. And then we enter into the next stage. So remember, there's anaphase A that pulls the sister chromatids apart. which were the kinetochores, and then there's anaphase B, which is going to be where the poles themselves are being pushed further away from each other. And then there's telophase. You want to think of telophase as the undoing or the opposite of prophase. So what happened in prophase? Remember, the nuclear envelope broke down. Well, what's going to happen? The two new nuclear envelopes are going to be forming around the separated chromosomes. So that's going to be one. The other thing is, you can see that the chromosomes themselves are starting to fade out in this drawing. That's going to be a way of representing that the chromosomes are starting to uncoil and decondense. Okay, so those are some major things that are happening here. So you can see that you're starting to see that purple nuclear envelope forming again, and it's going to capture all of those chromosomes that had been pulled apart. We can now call them chromosomes again, as long as they're still steamed. And then once they decondense and the nuclear envelope forms, you're not going to see them anymore. It's back to chromatin again. And now we have to separate the cytoplasm. So we've formed two nuclei. The entire cell has not divided yet. So this process is called cytokinesis. You want to think back that this process has started to occur as far back as anaphase. You can see that you're starting to form this sort of this furrow there in the middle. So the cell's starting to pinch off in the middle to form two new cells. So cytokinesis actually starts happening as far back in mitosis as anaphase, and then continues in telophase. You can see that cleavage furrow there. And then here's, you can see the cleavage furrow on either side here of these two cells in this micrograph. You can see the cleavage furrow there, another scanning electron micrograph. And so this process in animal cells, we're going to refer to it as cleavage, and then you have your cleavage furrow. And this is going to happen here in an animal cell. The animal cell can pinch because it doesn't have a cell wall. And what happens here is you're going to have all along the middle of the cell, around where that metaphase plate was, you're going to have these actin fibers that are along the surface of the inside of the cell membrane. And they're going to form this belt-like structure, and then the strands of the actin are going to be pulled tighter and tighter. So it's like if you wrapped a bunch of string around the middle of the cell, and then you're like pulling on either side of the string, and the string is kind of tightening more and more, pinching the cell off there. So you end up forming that cleavage furrow in the animal cells. Now remember, plant cells and other cells that have cell walls, instead, what's going to happen is you're going to form a cell plate instead in the middle, and then from that cell plate you build a new cell wall. So the cells can't pinch off in the middle because of the rigid cell walls that were there. Now keep in mind that in other types of cells that we saw with fungi and some other types of protists, that the nuclear envelope does not disintegrate away. And what happens to happen there is the nucleus divides, and then without disintegrating, the chromosomes get separated within the nucleus, and then the nucleus divides, and then the cell divides. So there's some differences with some of the other eukaryotes there. When we go back to the plant cells that I mentioned earlier, in that formation of the cell plate, that's a structure you need to know. Plant cells do not form a cleavage furrow. This is for an animal cell with no cell wall. For a plant cell with a cell wall, a cell plate is going to form first, and this image is showing this here. So what happens here is, let's say this is one cell over here on this side. This is the cytoplasm of it that's going to divide. So the nucleus is divided, so there's some new nucleus over here somewhere, and another new nucleus formed over here. And what happens here is that the endoplasmic reticulum is going to send materials, and those materials go to the Golgi, and the Golgi is going to send vesicles. And those vesicles are going to line up in the middle of the two cells, and they're going to start to fuse together. And as they begin to fuse together, they're bringing components of a new plasma membrane. And that new plasma membrane is eventually going to form there in the middle. And then eventually the cell starts producing new cell wall between the two plasma membranes. But this early structure right here that forms between the two cells, so there's one cell over here, and another cell, and it used to be just one cell. The two nuclei form, and then you begin to form that cell plate that's right here. And you can see all the vesicles are starting to fuse together eventually to form what would be the two plasma membranes, and then a cell wall gets produced in between the two.